All right, so hello everyone, and welcome to a session of Digging Deep with Quantitative Plant Biology, the webinar series. If you don't already know, Quantitative Plant Biology is an open access journal published by Cambridge University Press in association with the John Innes Center. We're delighted to be bringing you this webinar series thanks to our editors, authors, and the support of the John Innes Center. Today, we're pleased to feature Gita and Kedar, and they'll be speaking about their paper for quantitative plant biology. In a moment, I'll hand over to Olivier Hamat, who will introduce today's session. Before we get started, I just want to go over a few housekeeping items so you know how to participate in today's event. You've all joined in a listen-only mode, which means you'll be able to hear us, but not able to speak to us directly. However, you will have an opportunity to submit questions by typing those questions into the Q&A of the control panel. There's also a chat function as well, so you can enter questions in either of those. You may send your questions in at any time during today's webinar, and we will collect those and address them at the Q&A session at the end of today's event. If we are unable to get through all of the questions today, they will be included in a follow-up to the email, to the webinar that we send out later today. And should you experience any technical difficulties, please add those to the questions box as well, and a member of our organizing team will help or endeavor to help you. In the unlikely event we do experience any issues, we will message the audience and restart the webinar. So to summarize, you can submit your questions at any time, and you'll also receive a follow-up email at the end to know how useful you found it and any other opportunities to ask questions. Also, really quick before we get started, I want to encourage you all to check us out on X or Twitter. We are at QBB_journal. I'll put that in the chat as well. So give us a follow on there and feel free to start any discussions about today's event or any other things with hashtag QPB webinars. Just to start the event and start any meaningful conversations. All right, so to kick things off, I would like to invite Quantitative Plant Biology Editor-in-Chief Olivier Hamat to introduce today's session.
Thank you, Casey. Thank you all for being here. So today we are very happy to have Kedar and Gita. The paper that we're going to discuss fits very well with the scope of the journal. There's a lot of quantitative aspects. And I also want to mention the idea of time, actually, that is sort of underlined in the paper, because pollen is one of the longest lasting thing on Earth, actually. Palynology, that's where you see the pollen that is staying forever, right, as a dead cell, though. But it's thanks to sporopollenin that it's so resistant, right? So that's what it's actually, I think it's the only molecule that you can't digest, right? So that's why it's staying so long for on Earth. But the paper is about viability, which is, I think, it's something that we are all concerned about when we do experiments. I'm sure many of you are aware of Evans blue staining or PI staining to see if it's live or not. But here we're going to talk about the quantitative imaging approach, which I'm very happy to share with you today with this webinar. And Kedar and Gita, the screen is yours. Thank you for being here.
Share screen.
And here is the magic of the multi-screen thing.
Thank you so much for bringing us on there. There we go. Can everyone see our slides?
Yes.
So a very good morning to everybody in the West and a very good evening to my friends and colleagues joining us in the East. Along with Kedar and other co-authors of our recent paper, I'm very glad to join this session, Digging Deep with Quantitative Plant Biology, the webinar series. We specially thank the journal and the John Innes Center, Cambridge University Press. You, most of all, Olivia, for introducing us, as well as Casey, Julia, Alison, everyone on the entire QPB team for the support you've provided to us leading up to this day. I am Gitanjali, you can call me Gita, and here, right here with me is my colleague, Kedar. We both will be presenting the slides today, but all our co-authors are here with us. Suresh is on the panel with us as well, right here in the main panel of today's webinar. And our team, Abhishek, Nishant, and Vipin are in the audience. We look forward to having a lively discussion with you on how well botany and physics go hand in hand. The first thing I want to do is to begin with a short background introduction to the work we do in the lab at NIPGR. Some of our research interests are on the screen. We have a diverse, group of questions that we ask in the lab, spanning genomics, cheminformatics, algorithm development, as well as training in data science, genomics, as well as conservation. One of the major themes that I am very keen on is natural product biosynthesis and biosynthetic pathways. Our approach has been mostly knowledge and data driven. So we explore enzyme active site architectures, as you can see in the center of this screen, mainly to predict how subtle changes you see in the active site cavity lining residues may have a role to play in identification of substrates of products with great accuracy. So that's been the general story of where we've been going with research. But today we shall present some of our ongoing and yet very interesting work at the interface of physics, botany, data science, and invasion biology. So in collaboration with ecologist Suresh Babu at the Ambedkar University of Delhi, we've been investigating community diversity, habitat vulnerability, and many other aspects about the dominant invasive species, invasivity as well as invasibility. In the lab, it is my students, you see them on screen. Abhishek maps these phenotypic features to genomes and transcriptomes, as well as the metabolomes of the aggressive invaders. The current species that we've been keen on are Lantana camara that you see on screen, as well as Prosopis juliflora, which in Indian vernacular we call the vilayati kikar. There's also Prosopis, there's also Ageratum conizoides that you see on the screen out here. Now, the interesting bit is how this work was transformed completely by the influx of physics. So during the pandemic, I read about this guy, the optical physicist at IIT Delhi, who had done, Kedar had made this new invention at IIT Delhi, a low-cost microscope that he calls the computational microscope, using the quantitative phase information that is not directly measured, but actually estimated using numerical processing of hologram patterns at the detector plane of an otherwise simple microscope. When we first met, Kedar and I both spoke about the challenges of finding biological cells to apply. He was finding it difficult to find cells to apply his invention to, even though he had at that time, as you can see on screen, successfully identified sickle cell features in patients' RBCs as well as healthy RBCs. But he was stuck with, as we all do, approvals via hospitals and lots of paperwork that the red tape you need to get rid of in case of human studies. That was when I asked him, why don't you look at plant cells instead? And he said, which plant cells? And that is when Suresh came in and said, let's look at pollen cells, right? Now, Suresh had already been at that time discussing the correlation between ploidy levels and pollen stainability of different cultivars of the invasive Lantana. He's got colleagues in the American horticultural community, and that is where they've been showing, at the University of Florida, they had shown this correlation. And at NIPGR, we were trying to work out with different botany departments in the University of Delhi colleges to look at pollen. So then Kedar said, all right, let's take a look at the pollen grains in my DHM, but what on earth is the pollen? So for those of you like Kedar who are strangers to botany, pollen grains represent the male gametes of seed plants, and their viability is critical for sexual reproduction in the plant life cycle. Palynology, as Olivia just told you during our introduction, is the study of pollen, and you see, several pollen on the screen as well. It's critical. Basically, it can answer a huge number of questions ranging from botanical, ethnological, geological, etc. But recent work, as I told you, allowed us to reveal the importance of pollen viability as well in invasives, right? So that is where we got on and then said, all right, so what does the DHM show us? So then when Kedar started to look at the pollen grains, the question was, which plant should we look at? And of course, the discussion, who else but Lantana Lantana camara because we'd all been so keen on Lantana, the invasive species. Lantana pollen, as some of you who have worked with invasives or have looked at botany before, Lantana pollen now have this typical tricolpate feature, okay? So they've got these three colpi, as you can see the simple microscope view in the center in orange, if you can see my cursor. And this is the view that you usually get to see when you look through a simple microscope and identify pollen there. But also on the right side in black and white, you see the scanning electron microscopy image of the same pollen grain. It's far more resolved, highly resolved, and you can see more detailed features which have over the years assisted botanists in taxonomy, which means identifying the species based on their pollen. Now, what we wanted to use this for, and you see that picture on the left side, is to understand viability detection. And the traditional output of a viability detection experiment is when you stain the pollen and then you try to look at it with traditionally trained eyes, and you see them in colored and uncolored blobs. So as you can see on this screen over here, the dead pollen are the ones that did not take the stain. The live pollen are the ones that did take the stain. And now the DHM reconstruction, the DHM gave us a completely different view, to say the least. So what you see here is the bright field image of a regular pollen. It has taken the stain. We know it's viable because it's taken the red colored stain. The image plane hologram figure of the same pollen is the one you see in black and white in the center. And this is what you see here is a fringe bending pattern. These fringe bending patterns on the hologram are the immediate reconstruction of the microscope. And for a biologist like myself, what we saw, what was more surprising was that last figure, which you see is a kind of elevation on a 3D surface plot. Now, biologists like myself and a lot of you probably think about 3D surface plots by default as representative of altitudes in geography rather than molecular views inside a cell, right? But for physicists like Kedar, and I'm going to hand over to him now, it is a common practice to render objects as surface plots because the phase map represents optical path lengths. The surface plots provide a sense of depth information regarding the object that you're looking at. Already, this sounds like rocket science to me, and to save you the hassle, I will now hand over, of course, the proceedings to Kedar, who's going to explain to you the entire concept of the DHM microscope. Over to you, Kedar. Aage aa jao.
Thanks, Gitanjali. I'm going to give a simple overview of what these pictures actually mean and what is this microscope trying to measure in the first place. To begin with, we will look at a simple picture. We have all seen a laser pointer. And so let us say we send a laser pointer through a glass slide, just like a standard microscope slide, which is maybe 1 millimeter thick with a glass of refractive index 1.5. We all know that light goes through material like glass and it slows down. So if you this laser passes through 1 millimeter thick glass slide, it will slow down by about 1.7 picosecond, where a picosecond is actually 10 raised to minus 12 seconds. And in addition to the glass, if there was a transparent object like a cell on the slide, which is in the light's path, it will slow down further, tiny bit more, just about tens of femtoseconds more to cross the slide. And this time delay, which is really, really small and very hard to measure, is actually imprinted on the light beam by means of what is known as a phase change in the light beam. And the main idea of this DHM instrument is to precisely measure this phase change as a function of the XY coordinates along the slide surface and to generate what we call as a phase image that you just saw in the last slide. We will look at what does this phase mean with a slightly different view. So let's suppose we have this coherent laser beam and that passes through a glass and a cell on top of the glass. What happens is the coherent laser wavefront gets distorted because it has now passed through the cell. So different parts of the beam have actually seen different thickness and different refractive index of the cell. And so you get this 2D distorted surface that represents the phase of the light wavefront. This phase of the wavefront can be approximately written in this form. It is 2 pi divided by lambda, which is the wavelength of this laser beam, and it's an integral on the thickness dz of the space-dependent refractive index of the cell. So n here represents a space or coordinate-dependent refractive index, and n0 represents the refractive index of the surrounding medium that is surrounding the cell itself. And so you can see that the phase map actually represents a projection of the refractive index relative to the surrounding medium. Now, interestingly, while we have been doing imaging and microscopy for a very long time, a few centuries, everybody knows in optics that this quantity cannot be measured directly by any camera or a film or any other sensor at optical wavelengths. And so we need to convert these really tiny time variations into spatial variations in the form of a coded image that this DHM is trying to achieve. And this image of this function theta xy will be called as a quantitative phase image of the sample. Now, when you hear about phase, the first thing that probably comes to your mind is the phase contrast kind of microscope that many biologists actually use. So I want to differentiate between phase contrast and the quantitative phase here. So the phase contrast, the microscope in a bare bones form looks like this. If you have an illumination on a sample, then you have an objective lens, a tube lens, and a camera sensor. And behind the objective lens, or embedded within this objective lens, there is a Zernike phase contrast filter. And this filter actually provides an enhanced or modified contrast of this object as a visual aid. This image, you can see here on the right-hand side, if this is a bright field image, this is how the phase contrast will look like. If this is a bright field, tiny features that are not visible in the bright field suddenly appear in the phase contrast image. So this is a very good visual aid sometimes, but this image is actually not quantitative in nature. What I'm going to show you with this DHM, or digital holographic microscope, is actually a quantitative image. Now, if you want to see how we change the microscope to this DHM, this is how, there is a slight modification to the configuration of imaging. So you have a laser illumination. So this part of microscope is almost the same as before. There is no phase contrast filter. So it's just a normal bright field microscope. But this laser is first split into two, and then this beam passes as a reference beam through air, and then it is brought onto the camera again at a slight angle, and that creates these interference fringes. So this true phase map of this object on this sample slide is now converted to interference fringes that look like this. So you can look at the bright field picture, which you can record without this, the secondary beam or reference beam. But when you have the laser on, and when you have both the beams on, you can actually record something that I call as an image plane hologram. These lines that you see are actually interference fringes that everybody learns in undergraduate physics. Now, you see that whenever there is no cell on the slide, that part, the lines are really straight lines, undeviated straight lines. But whenever the light encounters this submedium, like a pollen here, these lines start bending. And this bending of these fringes actually contains information about this phase that can be recovered using some algorithm. So you can take this data as an input and recover this phase profile as an output. That is, we will call this as a phase image. Now, notice that in this in this modality, we are not really dependent on absorption of light or We are not dependent on something like fluorescence, which requires very high power illumination. We are just trying to send light through a transparent object. And so the laser power that is used here is really low in milliwatts. And that is very similar to the laser pointers that we use for presentations. So let's move to the next slide. I want to tell just a little bit about what is the innovation that we have here. Interference is actually a wave phenomenon, and it is really known a long time, meaning it's known over 200 years, and it's already part of standard undergraduate physics curriculum. And in fact, the basic Mach-Zehnder interferometer design that we have used is also very, very old, more than 100 years old. Now, what is new in our work is that while it is known that this interference record can be made, getting full diffraction limiter resolution from a single interference frame is traditionally considered to be a very difficult problem, because this problem is an incomplete data problem. So to tell you in a simple language, an incomplete data problem means that you are trying to solve something like for 100 unknowns when you have only 25 equations available. Now, this is really difficult to solve, but we were about 10 years ago, we developed a novel sparse optimization algorithm to solve this kind of problems. And once we were able to solve this problem in a practical time for an algorithm, then what it translated to was we could do these measurements, meaning less measurements, and that actually meant we could build a simple instrument by bypassing some complex hardware. And interestingly, this new math solution to an old concept, an old problem, actually led to a new microscope device. So if you want to see what we have been doing for the last 10 years, we did establish this single shot phase reconstruction algorithm in the literature, mostly physics and optics literature, when I had no idea about pollen. And but at the same time, we started building prototypes. So first prototype we had was on a breadboard-like setup that looked like this. It doesn't look like a microscope at all, but just a trial version. And that was embedded in a in a more initial prototype, and that already started giving us many interesting results. So we converted this into a more advanced instrument, which is now available in the central instrumentation facility at my institute, IIT Delhi. And it is interestingly started giving some publications for various groups in chemistry, and now a new publication here in quantitative plant biology. And but this whole concept is just about 10 years old, and it is based on a large number of series of articles that we published over the last 10 years to establish this single shot concept in the physics literature in the first place. But now that the instrument is ready, we are willing to and very interested in collaborating with groups within my institute or other institutes across India or anywhere, and see something interesting with this system. Now, let's come back to pollens, and to confirm, what we want to see in this pollen study. We first prepared the pollen sample in the traditional way with staining. And so you can see there are two groups of pollen that are shown, one on the left-hand side and one on the right-hand side. And you can see that these pollen on the left-hand side, they are confirmed to be viable because all of them have taken stain. But the image plane holograms of these pollen look like this. They all have this characteristic that the fringes actually bend whenever the image encounters, these lines encounter the pollen in the image. And all of them with this algorithm lead to the space profile, which is kind of a projection map of refractive index, as I said before. On the other hand side, if you look at the non-viable pollen, the shape of this phase is completely different. It's really flat. And in fact, you can see that inside these, inside the pollen region, these fringes are still quite a bit straight. They are not as much bending like in the case of viable pollen. So these phase maps actually indicate either that there is less material in the non-viable pollen, or that the refractive index contrast with respect to the surrounding is really very, very low. And so we are not getting any appreciable phase out of these images. Also, you can see that whether we have the dye or we don't have the dye, the uniformity of these images is, they are all uniformly bright approximately. This is because the dye that is used here for staining is known to have really low absorption at the laser wavelength, which was 650 nanometers. Now I will hand over to Gitanjali. She will explain a little more about what we did with these images.
Now, together, this is something we did together. Essentially, when the DHM, as you have seen already, is giving an image plane hologram, which is a black and white fringe bent version of the pollen with or without stain, the first idea that we had was, what is the need to do any staining at all? For the last 75 years or 70 years, at least, since this whole mechanism of doing pollen staining for viability was invented, all of the colleges, as you see, undergrad colleges, all of the undergrad text requires this method for students to be able to do, for any biologist or botanist to be able to understand pollen viability. So the question I asked and we thought was, if absorption, as Kedar has just told you, does not have a role to play in the fringe bending, then can we identify or detect viability in the absence of staining, a label-free mechanism? And the second thing that happens here, as you can see on screen now, is that the DHM returns to us more than 30 different mathematical features which are extracted for every single pollen grain. And these different features, we believe, these phase features, they're all phase features, right, Kedar? We believe that they may have a role to play to allow us in the long run to be able to predict, other features of pollen, such as, for example, polar diameter, the mesocolpium distance, equatorial dimensions, aperture size, spine diameter, exine thickness, you name it. We are open to trying and seeing whether this can be predicted at all. For now, for this work, just the viability part, we did not have to use all the 30 plus features that were mathematically extracted by the DHM. The only one that we used was the mean. As you can see here, we used the mean phase, and within the mean phase itself, whether the pollen is viable or unviable, stain taking or not, is easily distinguishable on the phase feature hologram as well, and much better on the 3D surface plot. We tried to do this with over 500 different Lantana pollen grains for setting up a preliminary proof of concept, which we published here in QPB. But currently, Kedar and I already have the wherewithal to expand this to thousands of pollen. And I mentioned earlier, and Kedar mentioned just now, you see the phase features also help us, give us a huge potential for machine learning. And we are particularly keen to explore whether, other traits of pollen or the plant health can be predicted by some of these phase features. So as you can see over here, the first thing, and I think Kedar, you should take over now, there are two very important bits that we see here. Yes. I'll come to the biology later. You explain from your
side. We were able to actually image about 500 pollen in one run, because really there is no sample preparation. These are freshly prepared samples right out of the plants, and then they are in fresh water, and there is no real processing required. Also, unlike something like SEM, no drying or metal coating is required, no staining is required, so we can directly see these pollen. And we can see that, if you try to look at their mean phase values, you can see that these are the non-viable pollen and these are the viable pollen. The viable pollen surely show larger mean phase value. And you can see that there is some overlap here. So Gitanjali, maybe...
the overlap region is something I find very interesting as a biologist. And Kedar, interestingly, because he records every single pollen data as an individual shot inside his machine, was able to, when I told him in this overlapping region, Kedar, let's see what are the pollens sitting there. Are they viable? Are they unviable? Are they in somewhere in the middle? What are they? So he was able to extract 4 individual cases from this overlap region, and you see the 4 individual cases on the screen right here. And as you can see, the first 2 are clearly signs of where the stain was not taken, and so therefore they are truly inviable. And the next 2, strangely, are a bit different. They seem to have taken the strain and yet they appear to be in the gray region or the dark region. So what I believe now is that the overlap region potentially is something new altogether, which we have not even considered in the past because we never had a way of measuring this. And so therefore these overlapping pollen may represent in between stages between viability and non-viability, which gives in turn a future research potential for us to be able to, because we are measuring every single bit of how much are they away from the two populations, the clear populations of viable and non-viable. We are in a position now to be able to say whether, sugar content of the pollen can be measured, hydration, could it be pollen quality, and so on. And the other thing we want to say over here very importantly is that because we are collating all of this data and it's an absolute, recorded feature set with us, the joint combination of bright field images as well as the phase images can help us create a huge catalog for the first time, an atlas of pollen grains for the world, which has not previously existed at all. And we believe that it can be an educational tool, particularly for undergrad studies as well as in grad botany. And this is something we want to begin, but right now we've just got a few thousand, it's a very small amount for which we need more of you to join us and collaborate with us so that this catalog can truly become an atlas of pollen grain data all over the world. Did you have something to say there, or can I? I really want to move into the fact that the DHM features, maybe just like mean phase allowed us to measure viability, Kedar, I want that other phase features, I want to check if they can provide other traits and give us a handle on any of these other traits that are listed here. And personally, for me, finally, what I consider the holy grail is the ever mystical Darwin's question of ploidy. Is there a way to be able to predict maybe ploidy by looking at these pollen? But we've not yet told you the label-free advantage.
Yes. So here on this slide, I just want to bring the real label-free advantage that you can have at least as far as the viable, non-viable question is seen. So you can see that this image actually represents just the hologram. We have not even recorded the bright field image for these pollen. There is obviously no staining. We are also looking at this at a low magnification. Instead of 40x, we are looking at 20x here. We can also go 10x, I think, without any problem. And you can see that now, if I just wanted to identify which pollen is viable and which one is not viable, we can just see that, this is non-viable because the fringes are actually not bending so much. Whereas in these other cases, these are indeed viable pollens because the fringes are bending. There are various shapes of that we can see here, but can almost estimate that this will create the same kind of phase values that are on the viable side. And so you can talk about...
the non-destructive sample preparation allows us to use this for several different downstream preparations, or downstream applications, not just in invasion biology, but also in crop agriculture, which we had not initially thought. To be very frank, initially, the three of us started this whole analysis as a blue sky idea with no immediate application in mind, you see. So Kedar, Suresh, and I, we soon realized that there is huge potential of such single cell analysis of pollen grains, not just for other globally important invasive species, but equally so in case of a large number of agricultural crops that we are now keen on investigating. I also see in the chat box, I have a parallel one with me. Lalita has asked a question about what are the traditional methods of pollen viability detection. You see them on screen, Lalita. Traditionally, for decades, we've been identifying pollen viability by manual staining in the lab and microscopy. And the holy grail for checking viability is actually having to germinate the pollen, which means for each individual pollen, imagine the amount of time you will take to actually germinate it into a plant. What we have devised through the DHM with, by bringing together botany and physics is an efficient, label-free, non-destructive method using digital holography. And the highlights of this method, as I hope to show in the next slides, are its scalability, its scope for wider applications in AI, and farming.
So the label-free here means that the one that we have identified as viable on the slide in a non-destructive way. If we can physically separate this pollen, we can also use it for further applications. So this is not possible with the stained pollen, because once it is stained, then it is not usable.
Ah, I see. Yeah, that's something really important. Okay, so I think this is our last slide. What we want to say is there are huge applications that we have for DHM in plant science. We are currently generating enormous. The first thing for me as a data scientist is that we are generating enormous amounts of data that can steer and accelerate research on development of different stages of these microspores to enable selection of specific microspores or pollen at appropriate stages. Now, for seed production, for example, many of you who have worked in agriculture would be aware that such knowledge could help optimize the female to male ratios. You could develop pollen dilution formulations. You could find or create new storage protocols, thereby saving not just time, but also space in the seed banking system, securing long-term stocks of very high quality pollen and so on. Another important issue to consider is where can we apply this to? We can use it on invasives. We've shown the proof of concept for invasive species. What kind of target crops can we use? A major feature in palynology is that pollen quality, pollen health is often controlled by abiotic features such as temperature, humidity, radiation, and many other genetic factors as well, which might be in breeding depression. You could have, direct and increasing negative impacts that inbreeding depression may have on pollen viability, on pollen quality, on pollen health, and many other things. And crops, certain crops, especially our major staple food grain crops, which have very short pollen lifespans, such as wheat, rice, corn. You see them on the screen over here. These could gain hugely with research via the DHM. Pollen quality is also a very important parameter in plant breeding. You could come up with better pollinator lines. And our goal now, therefore, as you see on the screen, is to build, our aspiration, I should say, our aspiration after this work that we've published in QPB together is to work towards a portable DHM pollen analyzer, or should I say with the added feature of cell sorting. If you analyze, this is a live pollen, this is a dead pollen, can you separate them and give me only the live ones? What I'm saying is to come up with some kind of a cell sorter. Kedar, can it be done, do you think?
Yeah, meaning there is options like microfluidics available that can be combined with the DHM, and that offers really great potential for going to all these features like portability, efficiency. It's a rapid kind of analyzer, which is label-free, accurate, and maybe we can do some sorting as well.
Can do some sorting. Hopefully, we'll be talking to you next time about having come up with a device that does the cell sorting as well. So in conclusion, what we want to say is that, one, the DHM can rightly distinguish viable and non-viable pollen grains. It's a new tech after 60 years of no innovation in the field, apart from returning more than 30 mathematical features that can lead to more ML and AI. And then the in-between space, the gray region between the neatly viable and neatly non-viable pollen are where I'm very keen on. What is that space telling us? What is that dark gray space got for us that we might learn from? The second conclusion is that our results can definitely be scaled up to enable label-free, distortion-less, stain-less analysis of hundreds already and thousands after cell sorting of pollen grains. And finally, I've said this already, there is potential for agri-farm applications, portability, and scalability. With that, I would like to just on behalf of all of us, thank you for joining us today, and special acknowledgement to our organizations, respectively IIT Delhi, AUD Delhi, the INAE, where Kedar is an Abul Kalam Fellow, and the NIPGR for me. Thank you also to you, QPB webinar series, for having us here today. We are very happy to take questions, if any.
Thank you.
Thank you very much. Thank you. I'm the only one clapping. Thank you very much for this lively duo, lively in all sense, actually. So there was even the Schrodinger's cat thrown in. So congratulations, great story. So there's a number of questions. So priority to questions from the audience. Let's see. So there's a question from Lalita Joshua. Has any similar study been done on other flower as well, flower species? How many species do you have?
Kedar invented this microscope 5 years ago. We used it on plants for the first time last year. We are the only people on the planet who have ever done something like this. I'm trying at NIPGR to get this machine here so that more plant biologists, and I already have a lot of friends and colleagues among Delhi, at least Delhi University college teachers in the botany departments, where we are hoping that we can bring in more flowers and more plants. Suresh, do you have any comments to give there? The answer is no other flowers have been tested, but Suresh may know what other species we might.
No, I mean, there is this particular publication, of course, is proof of the principle, essentially, that this can be done in this manner. I think there'll be a whole lot of work that is going to come up right behind this. We haven't scanned anything yet, anything other than Lantana yet, but naturally, there's a lot of possibility there. Yeah.
Yeah, actually, to follow up on this question, I was thinking that you could even extend to spores in ferns or even fungi, right? Possibly.
Yes, absolutely. As of now, Kedar can tell you better, our limit is about from 20 micrometers to about 45 micrometers. We don't want to go larger than that, and we don't want to go smaller than that. So if your target cell, can you?
Yeah, you can go up to 5 microns.
We can go up to 5 microns. So
Very easy.
Okay, so there's a big spectrum. All right, thank you. Okay, so there's another question from Lalita. What are the other mechanisms to check viability? Maybe you can say something about the Alexander Red and so other methods.
We've got different stains that we stain the pollen with, Lalita, and these are sometimes overnight stains, but all staining methods without question kill the pollen. So once you know it's live or not live, you can do nothing with it because you've basically finished it off. So that's a bit, that's where we wanted to be able to, and that's why the world is very interested in label-free detection of pollen viability. And the method that we've come up with does not require any kind of staining, and therefore we believe it's far more useful. Do you have any comments there, Suresh? That's it, right?
Yeah, no, I agree. Yeah, that makes a lot of sense. And the last question from Lalita, but I think you answered it. She was asking, do you compare with SEM? But I guess with SEM, you kill the pollen, right? So that's would be the same.
With SEM, there is another whole world of problems, Olivia. I mean, it takes so many days to get the SEM sorted, and you completely go blind trying to search for that single pollen. For working on a single pollen, gold plating it, freeze drying it. Of course, freeze drying means you're killing it, of course. But the kind of time that it takes to get that very high resolution, beautiful image, it's a lovely image, but that image eventually, it cannot be used. So
you can't scale up for sure. Yeah. So maybe I'll take a question from the Q&A. Anika Kayum is asking, can you please tell what is the concept behind the bending of fringes for viable ones? Is it pollen physiology or what?
This is a generic feature, so it has nothing to do with pollen. You can put a glass bead there. There also the fringes will bend. As soon as the light sees a different material with a different refractive index contrast and different lens, it will show a bending. This is just a coding mechanism to actually code these time delays that I talked about into space domain. And so it's a nice way to see things. We don't have electronics that can measure time delays of femtoseconds so easily, but here it is, you can just put it on a regular camera and you can encode that information in a different way.
Okay, maybe I have a sort of a follow-up question. It's not exactly the same question, but since you put your pollen in water, you might have some Brownian movement, right? So the pollen can move a little bit. And is this a problem or?
So that's where the single shot idea is great, because we take an exposure of about just a millisecond. So at that time it doesn't move that much, and we are able to reconstruct the phase after that.
Okay. Okay, let's see. So there are more questions. From Rupaksa, can this method be used to identify plant species?
Suresh would take this because he's had the biggest question from the beginning. Do we want to identify plant species with this? My personal view right now is not, but Suresh, what are you saying?
This is actually connected to the previous question, because the fringe bending has to do with structural elements in pollen. For instance, the material it is passing through. I mean, it is for is for Kedar to extend this. If it can go to pollen architecture, I mean, it can visualize. So pollen, each species of pollen has specific architecture, and if it is possible, right now we are at a very early stage, but as we go towards the library and so on, it is possible that some of these patterns can be standardized, and I think we will be looking forward, looking towards that. Probably we'll be working with Lantana pollen for some more time. If we can elucidate the structural elements to an extent, then I think we can go forward from there. So far the question I think is this is Rupak's question. Yeah, Rupak's question. Yeah, so far no, we're just starting out on this, but there is a lot of possibility here. The data that Kedar has milled out of this, we are not even using. What would you say, Kedar? The information we are using is like very small bit, is it not? For making it viable, non-viable, it's only very little bit of information we are using. There's a whole lot of material out there which is yet to be milled.
Low dimensional problem, I think
very low dimensional problem. Yeah,
that,
what we are answering is a macro.
Plant taxonomy has been fully worked out beautifully with the SEMs. We have beautiful SEM images in which we can completely identify the species, but SEM is very high resolution, requires all that time and gives so much more resolution. For us, at 20x, at 40x, you see a normal microscope. Kedar looks at even low dimension, 20x, and he's now wanting to go down to 10x. At 10x, you see nothing except the fringe bands. And so therefore, within the fringe bands, if the other features have something that they can capture about the identity of the species altogether, that would be a ceiling crasher, kind of thing to happen. But we've not looked at it. We can't till we have enough of a catalog created. That's why we want to create this catalog. We've just begun, as Suresh has said. Does that answer your question, Rupak, Olivier?
I think so. Rupa can say, but there's a lot of great, fantastic work in the Q&A. So just to mention, but okay, let's see. Very interesting. So yeah, there's one question about maybe needs some clarification. It's like you need to use a technique for a single pollen at one time. Is it too cumbersome? So I think but maybe you need to clarify the scalability and. Yeah.
No, we thank you for that question, Shehzad. This is not for a single pollen. We have extracted out single pollen to show you what it looks like. But when Kedar showed you the label-free advantage, he showed you that on a 10x, when you're seeing hundreds of pollen together, his DHM can capture the math to tell you on that slide with 100 pollen, how many are viable, how many not. So in one go, we can look at it.
Yeah, we are just cropping out a region around the one pollen to display these images so that all the details are seen nicely.
It's for the figure plots, yeah, but actually there's more data there.
Yeah, of course. So there's another question from Monica. Actually, it's more a comment. Congratulations to the team. This is wonderful and has a variety of applications. Before starting and pollination, manual pollination, this technique can help as one can ascertain the pollen used in ants pollination. This technique can also help understand microgametogenesis. But she is asking, one can use NMR to test viability. What do you think of that, NMR as an alternative? I guess more complex, but...
Yeah, meaning I cannot comment on it's really complexity of NMR is much higher.
Complexity of NMR is very much higher. Being able to use NMR is absolutely no comparison to the ease of use of what we have with single slides and a single microscope, which we can put into a little schoolroom also. So educationally as well, and insight-wise, at much lower cost and much lower tech, we can do much more analysis than as compared to the NMR.
Yep. Fair enough. There's another question from Kiran Prabha. Great work. Can this be used for plant sections for studying anatomical observations? Can you do it in tissues, in sections?
Okay, now this is a very interesting question. I'm not going to take it. Usually when we do tissue sections, we have to section the tissue. And some of you who are botany teachers may know how difficult or easy one takes to practice the sectioning of tissues. What Kedar does, lo and behold, is you don't touch, you just hydrate the pollen and keep it there. He's going to section it through his mat because the light is passing through the pollen. He can take out transverse sections without having to touch the damn thing. What do you do?
Yeah, meaning that's a research problem, I would say. Right now, we are not there to give you sections, but yeah, that's very much in the works right now to give you a tomographic image because this was initially, in fact, pollen, I think, is a great sample or a candidate to try some new ideas on tomographic imaging, which actually Nishant, who is an author on this, he's going to work on this problem. He's already started work on this problem. Yeah.
All right. Great. Yeah, nice prospects. Maybe I can ask another question that it's more like actually following up on digging even deeper into the tool. So here you're talking about viability. Can you extract variability as well? So if you compare different pollen, the type of patterns you get, have you started to look at I know, patterns that you see coming up often, or things that are a bit unusual, or I guess it comes down to your previous question on, extending the analysis. Yeah.
So the fringe bending is what we see. And if you remember the 3D surface plots, you generally see them very red, which means a lot of live information in the cell, meaning living cell, or darkness, which is bluishness, which means that there is a phase, the mean phase is very low, which means I don't want to say there's nothing there. We still don't know what the blue represents, but it represents inviability. To answer your question, there are lots of in-between spaces also. We believe there's data.
Yes.
Right.
Okay, cool.
Yeah.
There's another question. I mean, there are many comments say great work done, great, fantastic work. I mean, I just want to mention that. There's also a question from Manashwini Sambyal. As Gita mentioned, that this will have a lot of application in the undergrad studies. Is there any chance that students like us can learn how to use the DHM now?
Yes, sure. We are open to that. Yes.
So Kedar, as of now, the only device is sitting at IIT Delhi in the central facility, open for anybody to book and use. We are going to get a new device at NIPGR very soon. So there will be 2 devices right now, and we are more than happy.
There are some prototypes as well that are simpler.
Simpler prototypes. So
Cool,
then
perfect.
yes, we are very keen on connecting with colleges so that more students start to be aware of this and think of more ways this can be applied.
Very nice, very nice. Maybe I'll end, because I see it's almost at the end of the Q&A. Maybe one general question. So Kedar and Gita, you're a biologist and physicist. And so this is typically the type of work we like to publicize in QPB. I like to have this interdisciplinary work. But also I like to ask you a sort of a general question. So for you two, what was the most difficult in the collaboration coming from different fields? And what was the thing you preferred? What was, let's say, the highlights? Or in the process, let's say.
It's really nice because in some sense, our institutes are separated just by a road in between. And very little collaboration happens in this way. People are in their own silos. So first of all, it is really great to start this kind of collaboration. I think the students also benefit a lot to meet with other communities that are other than their own research work. Other than that, I think I don't think there have been too many difficulties. Somehow this one, we did not face communication gaps or anything. We just we had been talking about this, 3 of us, me, Gitanjali, and Suresh. So we just thought that this was a good sample to try, and somehow.
Okay, the difficulty was to try and explain to him, or to try and answer his question when he said, what is that red and blue? What is it that is taking the stain in the pollen? Now, I am a botanist, but not such a professorial botanist to know that the stain always goes only into the sugars or the DNA, and it does not be taken by the protein. I may have been mistaken, but then there were questions by other physicists, because we've been presenting this to the physicists community also, and their questions are so complex as to, you remember the questions your colleagues were asking, but they were valid questions, and that is where we both felt that we need far more data to be able to address questions like, what is it exactly that is getting stained?
The lot of interest from the physics people would be, what is exactly there inside a viable pollen and non-viable pollen? That the phase is so different, and physically, is it the index that is different, or is it the material that is different, or these are the questions that we they are our curiosity, and on the other hand, the applications may not require knowledge of all of that, but still it is interesting probably to know.
I find a very interesting comment by Professor Geeta Mathur. Thank you, Geeta ma'am. I think I'm going to get in touch with you about this. This is wonderful. So she says callus tissue can be converted to suspension and viability can be determined by that. So that's very
There you go. Yeah, I see that. So maybe I'll just read one of two comments. So Kiran Prava says it will be a real revolutionary optic asset for studying plant structure in relation to other parameters. This is from Kiran. And Bilal Hamadwani says, congratulations to the whole team. You people have developed important tool that has a lot of advantages over general use staining techniques. Well, people are very happy with the techniques. So I'm sure you'll have a lot of contacts after this webinar and after this article. So this is great. So thank you again for being here. I'll hand over to Casey and Alison.
Perfect. All right, great. Thank you all. So if there's no other questions, I really hope that this Q&A has covered a broad range of questions and answered anything you all might have had. I really want to take the chance to thank you, Geeta and Kedar, for taking part today and giving us this wonderful talk. It was fascinating. And Olivier, as always, thank you for attending as well. And to all of you attendees, thank you so much for joining us today. We will have another QPB webinar on November 30th. So please check out our QPB homepage. It's at cambridge.org/QPB, or check out our Twitter page as well. I'll put that in the chat just after this, and be sure to register for that one as well. But that's all. Thank you all so much for attending.
Thank you for having us. Thank you very much.